Hello, I am Dr. Gor Sarkisian. This is Biology 430 Neuroscience. And today we're going to be talking about the cells in our nervous system. We will focus on different cell types and of the nervous system, which will be including the neurons and glia. Neurons and glia as such is a very broad category. There are many different cells that are part of neurons and glia. These cells are different in structure, in chemistry, in function. But it's very important to be able to distinguish these two cells because their functions are fundamentally different in the brain and they both occupy a certain very important niche in the whole structure of the nervous system. Generally speaking, it is the neurons that make our brain a unique organ. Neurons are responsible for sensing environmental stimuli, communicate information from one cell to another, trigger physiological response to the environmental changes. Now the other cell type, the glia, they provide more of a support function, such as insulation, supporting newborn neurons, nourishment, protection. To study the structure of brain cells, scientists had to first overcome some obstacles, one of which is that neurons are in the order of 10 to 50 microns in diameter, which is 0.01 to 0.05 millimeters. So it is obvious that neurons, well, cannot be seen with the naked eye. Therefore, neuroscience, from the cellular perspective at least, could not proceed forward without the invention of compound microscope, which only happened in the 17th century. Uh, well, but still, even then, there were some difficulties. In order to look at something in a microscope, one had to make really thin slices of the tissue. And perhaps for some other tissues it was easier, but not with the brain. Brain had its own problems. First of all, these thin slices have to be in the order of about a diameter of a cell. But with the brain, which is more like a jello-like substance, it's very difficult to make thin slice in a tissue that is not hard. So scientists had to come up with a way to, well, harden the tissue without disturbing its structure. And for that, we had to make an instrument that actually does that. Now, in the 19th century, we came up with a process to fix the tissue. Fixation, for example, with formaldehyde, is basically a process in which the proteins are cross-linked with each other within a fresh tissue, and it hardens and preserves the tissue. And then, after that is achieved, the brain tissue would be placed in a machine called a microtome, which consists of a very extremely sharp razor blade, and with a machinery that can move a already hardened tissue at very, very small steps and cut a very, very thin slice. So with these tools available, we basically developed a new uh, field of study which is called histology. It's a microscopic study, and it's aimed at studying the structure of tissues. But well, why? Why? Because freshly sliced brain tissue is, for practical purposes, transparent. There is very little contrast between cells, and it's, for practical purposes, useless when we just put it under a microscope. So we had to come up with a way to solve this problem, and it was very elegantly solved by incubating the tissue in stains, in dyes that can selectively permeate certain portions of the cell. Okay, and today one of the most widely used stains was developed by a German neurologist, uh, Nissl, Franz Nissl, which was in the late 19th century. Basically, he developed this basic dye that would stain the nucleus, as well as some clumps of protein surrounding the nuclei of neurons. These clumps quickly started to be known as Nissl bodies, and the staining technique is till this day is known as Nissl stain. Nissl stain is very useful for at least two reasons. It is easy with the stain to distinguish between neurons and glia, and it allows efficient tool to study the arrangement of cytoarchitecture of neurons in different parts of the brain. Though Nissl stain is a powerful tool, it has some limitations in staining the whole or revealing the whole picture when speaking about neurons in the brain. It does stain the cell bodies, but basically nothing more than that. So another method was needed to be developed, and an Italian histologist, Camillo Golgi, derived this new method. In 1873, Golgi discovered that when a sliced tissue, a brain tissue, is soaked in a silver chromate solution, which is now known as Golgi stain, makes some portion of neurons become darkly colored throughout the whole structure of the cell. This revealed that neuronal cell bodies, the regions around the nucleus that is shown in the Nissl stain, is actually only a small portion of the entire structure of the neuron. Some of the early observations that the Golgi stain enabled included that neurons have at least two distinguishing parts. They have the central region, which was already seen with the Nissl stain, which is actually the nucleus, it contains the nucleus, and in addition, it has a lot of thin tube-like projections coming out of the central region. Now, the central region, which is the cell body that contains the nucleus, is called the cell body, but it's also known as soma. it's singular for somata, or perikaryon, or perikarya for plural. So these thin tubes that radiate from the soma are called neurites, and these are actually the axons and dendrites. Axons are generally long, they're uniform in the diameter throughout the whole length. Axons can extend for a long distance. And on the other hand, the dendrites are relatively larger in numbers, but they do not extend more than a millimeter or two. So the early neuroscientists um, observed that the dendrites come in contact with many different axons. So they concluded that dendrites must have a function of an antenna for a signal to be received by the cell. Using the technique that Golgi developed in staining, a Spanish scientist, Cajal, in 1888, performed a series of experiments and determined that the cells in the nervous system are actually standalone units, and they basically communicate with each other by contact. This observation or conclusion was in direct conflict with Golgi's approach, or Golgi's doctrine, shall we say. Golgi was he proposed that the cells in the nervous system in the brain, they connect with each other and they make this continuous structure, like the vascular system, like the blood system, veins and arteries. And that is known as the reticulum. However, Golgi argued the opposite. He argued that the cells communicate by contact. They're not continuous. So the idea of the cell theory, that basically cell theory states that all living things are composed of cells. These could be multicellular or single cell organisms. They are the basic units of life, and so on and so forth. So he argued that the nervous system also adheres to this cellular theory. And that interpretation is now known as the neuron doctrine. So basically, Golgi and Cajal were not very good friends. They argued till the end, even though in 1906, Cajal and Golgi shared a Nobel Prize in their work. They remained, shall we say, professional enemies till the end. So let's talk about a prototypic neuron. And let's begin with the soma. Soma, as you remember, is the cell body. It contains cytosol. It contains organelles, not unlike any other cell. It contains cytoplasm. And it has the same organelles that every other cell has. So from that perspective, it's no different than any other cell in our body. But wait, that's not all. One thing to note is that even though neurons, like any other cell, contain the same cytoskeletal fibers, microtubules, microfilaments, um, intermediate filaments, the cardinal difference is that in the neurons, the intermediate filaments are actually known as neurofilaments. It's more, it's for practical purposes, it's the same exact thing. So it basically reflects the structure in different tissues. That's all. Next prominent feature of a neuron is, of course, the axon. Axon is highly specialized, only found in neurons. Its function is to transfer information over long distances. The axon originates from a very specialized structure on a soma called axon hillock, and that region forms the initial segment of axon proper, which is the middle region of the axon. There are some differences between, obviously, the composition of the axon and the soma, and noteworthy ones include that there is no ER in the axon. There are no free ribosomes, or very few free ribosomes in the mature axon. The protein composition of the membrane of the axon versus soma are fundamentally different. And these distinctions, of course, correlate with functional distinctions also. No ribosomes, obviously no protein synthesis. The axons can travel from millimeters to several meters, over a meter. Axons often branch, and these branchings are called axon collaterals. And there are several different types of them. These can travel long distances also. They can communicate with different parts of the nervous system. Some occasionally can return to communicate with the same cell or to dendrites of neighboring cells. Such axon branching, in this case, are known as recurring collaterals. So the ones that return are called recurrent collaterals. So the diameter of the axon can vary from 1 micron to up to 25 microns. And as we will see later on in the course, because the axon is primarily involved in transduction of electricity, the thicker the axon, the faster the impulse travel speed is. Then comes the axon terminal. As the name suggests, that's the end of the axon. It appears as a swollen disc, and that part comes in contact with other cells or its final destination, and it passes the information to them, whatever the receiving site is, whether it's another neuron or muscle and whatnot. This area of contact is called a synapse. At the end, sometimes axons can have many branches. Each branch can make a synapse with many, cell bodies or dendrites at the same time within the region. These tiny branchings are called terminal arbor. So let's let's see, synapse. Synapse is connection of axon with another cell. The terminal arbor is terminal branching of axon. So when a neuron makes a contact with another cell, it is said to innervate that cell. It basically provides innervation. Innervation. So the cytoplasm and the axon terminal are very different in their individual compositions. Some of the things to note is that there are no microtubules in a terminus. No microtubules. There is an abundance of synaptic vesicles, and the synaptic vesicles carry many different chemical signals. And we will talk in depth about what's happening with the synaptic vesicles when an impulse approaches the synapse. There is an abundance of membrane-bound proteins there that are very, very highly organized and orchestrated in conducting this chemical signaling from cell to cell. And finally, this area has very large numbers of mitochondria. And I hope you can think of at least one reason why, or what this would entail to have a lot of mitochondria. And yes, that area is very highly energy dependent. A bit more on a synapse, even though we will devote a chapter or two specifically to study synapses, but at least some terminology. When two regions come together, the presynaptic and postsynaptic region come together, they form what's called a synaptic cleft. It is a tightly bound area that allows the information to be transferred from one cell to another through a synaptic transmission. That's a chemical transmission. So when an electrical impulse travels down the axon, it reaches the terminals and it triggers the release of chemical signal that crosses the synaptic cleft. Okay, these are neurotransmitters. And depending on the cell type, depending on what the chemical composition is, this chemical signal then triggers an electrical impulse in a subsequent cell. So neurotransmission is basically conversion of electrical signal to chemical signal, and then subsequently that chemical signal is converted back into electrical signal in a secondary cell. So This is essential for many of the brain computational abilities. Um, this is suggested to be a mechanism of memory and learning. Synaptic transmission dysfunctions account for many mental disorders. Many of the drugs, both medically used and recreationally used, are act on this machinery of chemical signal transduction. Next, we have to introduce axoplasmic transport. As we have noted earlier, axons have no ribosomes. Therefore, they cannot synthesize protein. So proteins must be synthesized somewhere else in the soma and shipped down the axon. And in the mid-19th century, Augustus Weller showed that a neuron cannot sustain itself if it's cut from the cell body, and it dies. And it's basically that degeneration of the axon is a result of the normal flow of the material from the soma to axon is terminated. It's cut. So the movement of material down to the axon is generally known as axoplasmic transport. So in the 1940s, American scientists did a very interesting experiment in which they basically tied the end of an axon, and they noticed that material started accumulating at the knot. When the knot was removed, the material continued moving down the axon at about 1 to 10 millimeters per day. Well, okay, it's good, but let's consider 10 millimeters a day. For long axons, that would take about half a year for material to reach the end. So through some other experiments by injecting radioactive amino acids into the neuron, they were able to determine that there are some fast axoplasmic transport mechanisms that allow about a meter a day transport. And these transports are provided by a motor protein called kinesin, and this is the transport from the soma to the axon. The anterograde transport. But we also have the opposite occurring also, in which material moves from the axon to the soma. It's basically the same process, but it's carried out by another transport protein called dynein. And I'm sure you have heard of this, you know exactly what it is. And obviously in both cases, ATP has to be hydrolyzed to allow this motion to occur. Next feature of a neuron are the dendrites. The dendrites are on a receiving end of the neuron. A single neuron has a vast number of dendrites, and the collection of these dendrites are known as dendritic tree, which is quite congruent with its name because the name dendrite is derived from the Greek word tree. So as such, the dendrites can be seen as the antennae for neurons to receive signal from adjacent axons. So when an axon comes in contact with a dendrite, we have basically the membranes of these two cells coming together. And when we talk about the synapse, as we earlier described, we have this two membranes that are in contact with each other, And if we look at that cleft between the synapse, we have a membrane on the receiving end, which would be the postsynaptic membrane, and a membrane on a transmitting end, which is the presynaptic membrane. So presynaptic denotes a membrane on an axon, postsynaptic denotes membrane on a dendrite. So the membrane on a dendrite, the postsynaptic membrane, is very rich in specialized proteins that respond to a certain neurotransmitter that is released um, upon receiving electrical impulse. So these receptors are called, these proteins are called receptors. And when they bind their specific ligand, specific neurotransmitter, they perform a very specialized task in the cell. Now, because dendrites can make many connections with many different axons, some dendrites have specialized structures which are known as dendritic spines to enhance the capability of making connections with adjacent cells. After the cells of the neuronal system were able to be visualized, especially after the development of the Golgi stain, scientists started classifying the neurons. And some of the most common classifications are as follows. The unipolar cell is basically a neuron with a single neurite that is extending from the cell body. If there are two neurites, the cell is said to be bipolar. And if there are three or more, then it's a multipolar cell. So unipolar, bipolar, multipolar. The dendritic trees can also vary from one neuron to another, and these are also classified and have been given some extravagant names like double bouquet cells or chandelier cells. There are other cells that are a little bit more down to earth, like alpha cells. And some of these classifications are unique to a particular part in the brain. For example, in a cerebral cortex, there are two broad classifications of cells, and they are the stellate cells and pyramidal cells. One can go further and classify these as spiny, meaning, for example, pyramidal cells that have dendrites that have spines on them, or aspinos, which would be in this case pyramidal cells without spines on their dendrites. The cells are also classified in terms of the connections that they make. For example, primary sensory neurons, motor neurons, and interneurons. We will talk about all of these and what exactly they do. They can also be classified based on their axonal length, Golgi type 1, Golgi type 2, which are local interneurons. And they can also be classified by the neurotransmitters that they emit, like cholinergic neurons that release acetylcholine in the synapses. Acetylcholine is widely common in neuromuscular junctions, and we will talk about what those are also shortly. Now let's talk a little bit about glia. And let's begin with a prominent glial cell astrocytes, which is a glial cell and it's about 20 nanometers in width. It's they are the most abundant glia in the brain. They fill up um, the space between neurons. They can control cell growth, neuronal growth. They can regulate chemical content of extracellular space. Astrocytes can envelop synaptic junctions in the brain and restrict the diffusion of neurotransmitters. They can remove neurotransmitters that have been released outside of the cleft. Um, these cells can also control extracellular concentrations of some substances that are very important. For example, these cells, the astrocytes, can regulate the concentration of potassium in extracellular fluid. And as we will see later on, potassium is very, very important in proper concentrations of intracellular and extracellular potassium is very important in proper cell signaling by neurons. Another prominent group of glial cells are myelinating glia, which include the oligodendroglia or oligodendrocytes and Schwann cells. They are very similar to each other in their function. Oligodendroglia is in the central nervous system, whereas Schwann cells are in the peripheral nervous system. Their function is basically insulating the axon. As you can see on this picture, we have this concentric rings of myelin sheath. This is actually a single flat cell. Let's say this is the cell body. It's basically something like this that wraps its membrane around an axon, and in this case this would be the axon, this would be the wrapped up oligodendroglia around the axon. So the initial studies done in Boston University by Alan Peters, who was one of the pioneers of electron microscopy, he showed that the myelin, actually the sheet that the cells that wrap themselves up around the axon, the sheet that they lay is actually made of myelin. Myelin is basically produced by individual cells, and between each cell there is a gap known as node of Ranvier, and that's very important, and we will talk in detail why. But briefly, myelin is electrically neutral, and we know that neurons conduct signal through electric potential. So basically what happens, an electrical impulse would originate here and then would immediately jump here and then here and so on and so forth. And if there is a demyelination happening, a deterioration of oligodendrocytes, that results in disorders such as MS, multiple sclerosis, and many others. Of course, there are many other types of cells in the brain, ranging from blood vessel endothelial cells, which are the cells that line blood vessels. There are immune cells such as phagocytes that can protect the brain from invading organisms. There are cells that are called epidermal cells, epidermal cells. These are basically, they line the fluid-filled ventricles within the brain, and their role is to direct the migration during brain development. The second class is, microglia. Microglia in this case refers to the phagocytes, which are part of the innate immune system. They can remove debris by dead cells and so on and so forth. They can take care of the degenerating neurons. Microglia may also be involved in some physiological functions too. They can potentially be involved in remodeling synaptic connections by gobbling them, which opens up a very interesting possibility of using these mechanisms for certain disorders. Now, just to recap, um, we have observed structural correlations with function. And obviously, we've seen that structural characteristics of a neuron tell us a lot about its functions. Neurons, for example, are cells that have soma, axons, dendrites, and create synapses. Um, for example, nestle stain or larger level of nestle staining means increased protein, um, which suggests specialization of some kind. The elaborate multi-branched structure of dendrites hints on their function of receiving many signals from many different regions of the brain from many different cells.